All right, hello and welcome everyone. I'm your host, McKenna Campbell, and I thank you all for joining us for this evening's presentation, Synthetic Medicines Are Not Black Magic. Throughout the event, please feel free to send questions to the chat box, and we will have a Q&A at the end. Before introducing tonight's presenter, I would like to share a little bit about the association. ACS is an international association of healthcare professionals on the forefront of cannabinoid medicine. Founded by physicians, ACS is uniquely positioned to advocate for the highest standards in the practice of cannabis medicine. We are an advocacy organization striving to guide lawmakers and regulators at every level of cannabis control. We also prioritize inclusivity and value having members from all walks of life, which motivates us to provide donate what you can memberships to qualifying groups. Additionally, student memberships are free, so please contact us directly if you have any questions about how you can get involved. On this next slide, you can see there are many benefits to joining ACS. For all of ACS's constituents, we provide evidence and experience-based education and resources to support the premise that cannabis should be treated as a medicine. Certainly feel welcome to reach out to us with any questions about membership opportunities. For our student leaders, we encourage you to learn more about our Student Ambassador Program. ACS is a nonprofit organization, and so we do rely on donations to continue our work. We appreciate any level of giving, and you can support us directly through the QR code or website link as listed on the slide. And Kelsey, if you could throw that link in the chat, that would be super helpful. I am now delighted to introduce Jason Kingsbury, Ph.D. Dr. Kingsbury is the Principal Investigator and Chief Scientific Officer of Citrochem Corporation, a new life sciences startup focused on flavonoid and terpenophenolic natural products as medicinal agents. He was an NSF Fellow during doctoral work at Boston College, where his fascination with donor-acceptor diazo compounds began. His thesis work culminated in a development of chiral bidentate and heterocyclic carbene ligands that have since shown utility beyond the boundaries of enantioselective ring opening metathesis. Dr. Kingsbury's NIH-sponsored postdoctoral work at Harvard involved total synthesis of members of glycolic diterpenes, a class of bioactive natural products holding 930 bicycle carbon frameworks. Starting his teaching career in 2006, his laboratory received BC Startup Funds and an award from the ACS Petroleum Research Fund. In 2016, he was a lead technical advisor in efforts to screen a library of biocompatible fluorophores in collaboration with scientists at the University of Washington. In 2019, he tested intramolecular carbanoline reaction with nerol, an isomerized form of the monoterpene citrol. The product of this reaction, isopiperidinol, is being exploited as a novel intermediate to facilitate the large-scale production of minor phytocannabinoid acids and menthol, a refreshing agent. Dr. Kingsbury has written numerous publications and standard operating procedures, including building timelines for routine safety audits of all research active laboratories and setting safety standards for compliance at Boston College, as well as authoring new SOPs for proper handling, quenching, and disposing of pyrophoric, alkyl aluminum, and alkyl lithium reagents in the wake of UCLA's tragic embuthylium fire. And without further ado, I'd like to turn it over to Dr. Kingsbury, and we can get going on his presentation. Thank you very much.
Thank you, McKenna.
Of course.
Let's go ahead and screen share. Okay, welcome everyone. I'm really excited to be here and really guide a discussion around our term synthetic that is an adjective of the noun synthesis. And I think it's no secret that we live in a world where there's biases and stereotypes. And for many of us, when we hear the term synthetic, we immediately think fake, artificial, somehow less effective than the real thing. And so we want to craft a better discussion around what that means and some of the opportunities. You heard a little bit about my background, and I'm a champion and a longtime student of organic synthesis as a practice. And I really feel it has an enabling power for confronting challenges at the frontiers of modern medicine. And so my goal in leading our discussion tonight is really to, first of all, focus on our own collective awareness of all the terms and buzzwords around synthetic and synthesis, and also to do what we can to demystify or destigmatize this word synthetic. And along the way, we will certainly go down some tangents, and I have a number of examples in which a laboratory synthesis might have been misguided. It might have been motivated for profit with little or no concern for how it might affect an end user or patient. But at the same time, we want to understand that synthesis is a complement to natural products chemistry. I'm actually a natural products chemist in terms of my origins. And I believe that natural products chemistry and plant-derived medicines really are enabled by synthesis as a science in a way that is synergistic and cooperative. As we mentioned, or McKenna noted, this is going to transition right into a Q&A session. So I'm actually going to make a few acknowledgments at the outset here. The first is I want to thank McKenna, Jordan, other members of the ACS for the invitation and this opportunity tonight. And I also want to acknowledge that there are legendary researchers who came before us, sort of setting standards. And one that I continue to draw inspiration from is quoted here on our first slide, Professor Raphael Mechoulam. We're just now past the spring 2023 passing of Dr. Mechoulam. And in honor of his contributions, I thought I would just point out, in the context of making these slides, I came across a YouTube documentary produced by Zach Klein, known as The Scientist. And this is a wonderful storytelling experience on the life of Professor Mechoulam. And this is what he opens with in the first minutes of the documentary. He basically admits that we have at our disposal, right at our fingertips, a group that is broad and diverse in terms of a family of compounds, and they seem to be modulating and interacting with an entire family of protein receptors in the human body, the so-called endocannabinoid system. And this system is of primary importance in so many of our day-to-day aspects of everyday life, from sleep to appetite to mood to our own immune system. It's proving to be an orchestration of small molecule interactions with an entire family of fairly involved and intricate receptors. And the idea is, let's just continue to do what we can as individuals and as practitioners to push it forward. Let's move some of these molecules and novel derivatives thereof into the clinic for the benefit of mankind. As McKenna mentioned, I just had a dramatic career shift. I was teaching organic chemistry at the college level for nearly 2 decades. One of the things I also wanted to share on this opening slide is another resource. It's NOVA's program called Forgotten Genius. It chronicles the life of Dr. Percy Julian, and by all accounts, Dr. Julian was, if not the first, among the first African Americans to receive a chemistry Ph.D. And he did so in an environment that was predominantly white and highly discriminatory. So this is also another great documentary showing the trials and tribulations, and it serves as a way to show how Dr. Julian was a role model for a number of underserved students and minority populations, particularly those interested at the forefront of medicine, this interplay between chemistry and biology. So for those who might want a little bit more exposure to organic synthesis and what it can do outside of the realm of cannabis, this is a great starting point. I need to be in my computer, right?
Yeah, you gotta get to that. You can probably click on it with your finger, the second slide on the left.
All right, so our agenda tonight is going to be pretty straightforward. We're going to go over these definitions I mentioned and move quickly from there to a graphical depiction that will carve out the relationships and the differences and similarities among phyto, endo, and synthetic cannabinoids. We're going to do what we can to point out their origins and some of their features, both good and bad. And we're going to also transition to how cannabis plants are themselves mini chemistry factories or chemical manufacturing plants. I'll touch on why some of these natural products might have been there from an evolutionary standpoint in the cannabis plant, in terms of their beginnings and their place in our history. And from there, we'll quickly move to how cultivators are moving to deliver finished products, plant extracts via extraction, processing, and purification techniques. Along the way, what we're going to be doing is talking about how fundamental science using physicochemical and mechanical methods are inherently capable of leading to unwanted outcomes. So there are degradants and contaminants that can arise unless we're watching carefully and monitoring our processes very carefully. And they will ultimately cause harm to patients and consumers if left unaddressed. And so from there, we're going to transition to a case study that is really the poster child for botanically sourced medicine. It's the story of the blockbuster anti-cancer agent Taxol. We'll go all the way to the discovery of Taxol, some of the challenges associated with its commercialization and delivery to very sick and solution-deprived cancer patients. And from there, we're going to talk about how the concept of semi-synthesis, the merging of natural synthesis with a few deliberate or purposeful laboratory steps, ultimately was a solution that in and of itself delivered clinically meaningful, impactful amounts of Taxol so that it could become the blockbuster drug that it is. And in the case of Taxol, semi-synthesis was the solution. And we're going to also relay some of the relationships between semi-synthesis and what we might call biomimetic chemistry, which is more or less a way of thinking about approaches to accessing these natural products or natural product derivatives in a way that is cost effective and mirrors some of the well-known reactivity principles that are already inherent in plant biochemistry. Let's dive right in. So I mentioned some definitions at the start. Synthesis is broadly defined as the step-by-step buildup of a chemical substance from a more simple compound, something we also often call a precursor or a starting material. And that is the stepwise action of chemical reagents or catalysts on exposure to that simple starting material. When we tack the term bio onto the word synthesis, we're simply referring to the production of a chemical substance inside of a living organism. Turning to the adjective synthetic, this is just of, relating to, or derived from chemical or biochemical synthesis. And very quickly on this first set of three definitions, I have a couple visuals to help us here. Outside of the context of chemistry itself, synthesis occupies a very high point in something known as Bloom's taxonomy. Bloom's is something that I often share with my chemistry students. It is a hierarchy that describes how we as humans go about higher level thinking and cognitive analysis. So at the low levels of Bloom's taxonomy, we have remembering information, understanding a context for its authorship or presentation to us, and it moves further to applying and retaining that fundamental knowledge. And as we move higher on this Bloom's taxonomy, the activities that we engage in become increasingly complex and multifaceted. So when we speak about synthesis or creating, it really bears the idea that we are going to need to be strategists. We need to think that along a given pathway or route to a desired outcome, we're surely going to arrive at pitfalls or traps that may be unforeseen to us. And we need to be anticipating them and also thinking about their solutions or ways around the roadblock before that we might even arrive at them or know that we've arrived at them. And that idea of amassing and synergizing all that you know, taking all your resources forward, is really what celebrates synthesis as its own thing outside of chemistry. The second photo here is of Scotty Kilmer. I don't know if anybody knows or recognizes Scotty here in this photo. He's jumped into the truck of one of his vehicles. He is a YouTube sensation, and he's 57 years and counting as an auto mechanic. I'm a do-it-yourselfer at heart, and Scotty Kilmer was the first one that really had a great explanation for why we use synthetic oil that costs more than traditional forms of motor oil. So the idea is we have synthetic petroleum-based products that are resistant to higher temperatures, they're more long-lasting, they protect our engines better, and they let us go further between our oil changes. And so the first two takeaways from this slide is that synthesis is already happening in nature, and we can often get to very favorable, superior outcomes in some cases by developing our own compositions of matter, or even mimicking or replicating what we see in nature. Two more additions to the list here. Phytochemical is any chemical compound that's found naturally occurring in a plant. So we attach that phyto descriptor on the front end. And I've mentioned or alluded to semisynthesis. This is where we merge any natural biosynthetic process with one or more additional laboratory or man-made deliberate protocols. And we bring the best of both worlds together. We often arrive at a workable, practical solution that would not be a standalone solution if we didn't have innate knowledge of how the plant or microorganism is doing its own synthesis. And I'll touch more on that later. I also have a couple definitions to carve out around our own physiology and how these molecules are interacting with proteins in our body. So the term cannabinoid is derived from the name of the genus of the plants, but it's also specifically a name for any compound that elicits a physiological response by interacting with one or more G protein coupled receptors or GPCRs. An endogenous cannabinoid is something that we have natively in our own body that is the native or endogenous substrate for one of these GPCRs. And the endocannabinoid system is the ECS for short. It's the superfamily or large class of membrane-bound proteins. So these GPCRs all share some sequence homology in terms of their amino acid makeup, and they are all conserved in having 7 transmembrane loops. So there are 7 loops to the protein structure that are embedded in the cell membrane. And then they have tails, one exiting the cell into the extracellular matrix and another on the interior of the cell. And what happens is a small molecule binds outside, causes a conformational change that ripples through or translates through the cell membrane and into the inside, and it induces an active signal transduction event, which is essentially thought of as an on-off switch leading to some inside the cell response. So among this family, we have CB1 and CB2 receptors. CB1 receptors found predominantly in the central nervous system, that's the brain and spinal cord. CB2 receptors predominantly in our peripheral nervous system, more localized in immune cells, but also a few other areas outside of the central nervous system. More recent research has identified an orphan receptor termed GPR55. I actually just learned this myself. An orphan receptor is one in which we do not yet know what the native substrate is in the human body. So we actually don't know what the normal or typical small molecule is that is binding in the active site of that protein. But what we do know is that these endogenous molecules or phytocannabinoids are also hitting on this same active site with either antagonistic or agonistic effects. There's also PPARs defined here and a TRPV1. The latter is actually an ion channel. So it's also a membrane-bound protein that functions as a pore. So by binding a small molecule, we can either open or close the pore, which controls the influx or efflux of metal cations. This type of diffusion in or outside of a cell is the basis for signal transduction and neurotransmission events. Lastly, we understand now that there are enzymes unrelated to GPCRs, but they're responsible for both making and breaking down endogenous cannabinoids. And these have become an additional target for drug design, a way to intervene with the endocannabinoid system. What we know is that the level or the concentration of endogenous cannabinoids is typically a sweet spot, right? Too much of one might lead to adverse effects. Too little, again, there's a sweet spot for concentration. And so there are enzymes whose predominant role, sole role, is to control the expression and maintain appropriate physiological concentrations of these small molecules. All right, where we are at the moment is past these definitions, and we're going to now look at a graphical representation that will carve out a better understanding of the differences, yet similarities in these small molecules. So the first two that are going to come up need no introduction. There are major cannabinoids, delta-9 THC, the primary psychotropic component of cannabis, as well as cannabidiol, which is very similar in structure, but lacks the third ring. And my own innate fascination with CBD is at the level of how lacking that third ring gives the molecule a lot more conformational mobility and freedom over its shape in terms of it interacting with completely distinct receptor targets than the delta-9 THC. And to the right, you see the prototype endogenous cannabinoid known as anandamide. This is a very simple amide functional group or functionality that merges ethanolamine with a fatty acid known as arachidonic acid. And so I find it fascinating that the natural products in essence, are mimicking the behavior and the binding, the affinity of naturally occurring endocannabinoids, primarily as a function of this very lipophilic or fat-loving, water-insoluble carbon chain that you see extended in anandamide and prominent in our major cannabinoids. Some buzzwords that are associated with these would be natural products, secondary metabolites. It's important to realize these molecules are already have already undergone years of evolution, and they by all means are safe from the standpoint that our receptors are already amenable to receiving their chemical information, and we know that they're metabolized well in the human body. I didn't do the, okay, there we go. Now, on the topic of synthetic cannabinoids, we have 4 representatives that were deliberately chosen to sort of build a story here. The first thing that I want to stress is, again, the initial development of these molecules was not to induce highs in the user or euphoria. We had fundamental, often academic-based researchers who were using these synthetic molecules to actually uncover and identify their protein receptor targets. So DWH is named after John Hoffman, who was at Clemson University. And this first molecule was later the subject of a number of bad actors performing unsanctioned or illicit syntheses of this small molecule and then marketing it to minors in the form of K2 or spice. But from a science perspective, Hoffman was actually targeting something that was easy to synthesize and would perform structure activity information around it binding to a receptor and allowing us to identify that receptor and ultimately do genetic sequencing and learn more about its structure. As we move to the right, we have another contribution from Professor Mashalom. This molecule looks more like the natural phytocannabinoids. The main modifications include a heptyl side chain that's been dimethylated and a position 7 hydroxyl group at the top of the molecule. And so the idea in the development of HU308 was, let's synthesize and test a metabolic byproduct that we know is more stable in the human body and potentially even more effective than, for example, delta-9 THC itself. So those types of deliberate modifications of functional groups were warranted in our motivation to find and identify improved medicines. I'm going to skip the third and move to AM 10257. Professor Alexandros Makrianis is at Northeastern University. This fourth example of a synthetic cannabinoid was a scaffold related to the indole nucleus you see in JWH-018, but again, a nitrogen-based molecule, easy to synthesize, cost-effective to scale up. And that molecule is a CB2 agonist that ultimately served as a co-crystallization development that allowed us to get a fine, high-resolution X-ray structure of the CB2 receptor. And then there's a wonderful example of a radioligand here in the form of the Pfizer molecule CP55940. So I have highlighted in blue that the hydroxyl proton has been substituted for with tritium. Tritium is a radioactive isotope of hydrogen. That means it has additional neutrons in its nucleus. And Pfizer made this compound administered it in model studies to animals where it was a potent CB1 agonist and bound in the active site to activate that enzyme, turn on signal transduction. But once we then fed those animals with other well-known cannabinoids or cannabinoid compounds, they displaced the radioligand. And by following the concentration and the very predictable time loss or time decay of radioactivity, it served as the identification that there was an actual endogenous receptor for these molecules. All right, so let's move to how cannabis plants form secondary metabolites and why they might have been there in the first place. So this is a cartoon that depicts the chemistry happening in glandular trichomes of particularly the fluorescences or the flowering buds of female cannabis plants. And this is very schematic in representing a building block from two different biosynthetic pathways, one a polyketide known as olivetolic acid, and another from the terpene pathway, are essentially merged at the 50-yard line, brought together to make CBGA, cannabigerolic acid, which is the mother of all cannabinoids. All other minor and major phytocannabinoids are derived from this core building block. How that happens is a lesson in the existence of multiple enzymes in the plant. I mentioned the acid form of CBG. It turns out that the acid comes first in all of these end products. What happens is a terpene cyclase in each of the three series is responsible for wrapping that 10 carbon side chain into an enzyme active site. And through subtle differences in each enzyme's sequence or ultimate tertiary structure, we get these variable divergent outcomes in terms of structure. Moving further down in the diagram, we can see that the typical mode of inhaling or vaporizing the dried plant matter is a high temperature process associated with us combusting the material. And that combustion leads to loss of carbon dioxide or decarboxylation. And so CBC, CBD, and THC each derive from their corresponding acid form. And we're going to use this as a segue to discuss how a lot of those compounds are further susceptible to ambient atmosphere conditions that involve exposure to sunlight, in particular ultraviolet rays or near UV rays, and also oxygen, which is an everyday oxidant associated with oxygen in the air. And the discovery of cannabinol marked the very first phytocannabinoid to be discovered by Roger Adams in the 1940s. It could be argued that these are themselves phytocannabinoids that derive from a metabolism that is unpredictable, but at the same time unpreventable. And I'll talk about the time course of some of those downstream transformations in just a moment. So we have this entire library or plethora of compounds that are structurally related and influence our endocannabinoid system in ways that we're still learning a lot about. And we have cultivators delivering products either in raw plant form or subject to further extraction and processing techniques. And one of the things that I'd like to do is sort of drive home that this is, an undeniable first course of action in developing new medicine, where we really need to structurally characterize and purify single molecules from the mixture of chemicals that come from a full or broad spectrum extract. And we go about this in two ways typically. We have physicochemical extraction where a solvent, whether it's an organic solvent like butane or liquefied carbon dioxide, those are fairly nonpolar. We can move in the direction of higher polarity with ethanol, which is arguably the least expensive of those solvents. We can also talk about solvent-free water-based processes. Water technically is still a solvent, but the goal there is if you have cannabis compounds that are water soluble, you might not actually later need to worry about removing the water. You can directly, use the stock solution as is. Without solvent, we have mechanical options. In the upper right, I show a photo of a rosin press. This is where high temperature and heat and pressure is applied to two metal surfaces that are clamped together and directly compress plant material and yield an oil that's free flowing and essentially represents a concentrated, highly usable, neat form of active phytocannabinoids. If you think about this moving to a very large scale, the picture says 1,000 words. All of these processes can be highly controlled and deliver the best case scenario in terms of final outcome, but we are at the mercy of batch to batch inconsistencies deriving from the plant inputs or the plant matter itself. So when it comes to considerations for creating a medicine, we have to understand that there are painstaking and time-consuming endeavors or protocols that we're going through to get finished form broad and full spectrum extracts or even single isomer cannabinoids. And we've got to worry about the things that those cannabinoids touched along the path to the final product. We worry about residual solvents or other contaminants that might be ultimately harmful to the patient if not rigorously removed. Okay, so I'm going to move a little bit quickly through this because it's some chemistry associated with some oxidative and photochemical processes. But just for the benefit of our listeners, delta-9 THC is highly susceptible to air oxidation, leading to that cannabinol and the delta-8 isomer of THC, which is actually the thermodynamic or energetically preferred position for that double bond. I've ranked these molecules in terms of their known psychoactivity. Delta-9 THC is still the most psychoactive, but we worry about users who might be consuming CBN or delta-8 products when in fact those are newer to market and have been less studied in terms of safety and efficacy. The basis for something called a Beams test is actually what allows certain on-the-fly practitioners to identify whether you have a hemp-derived or a phytocannabinoid product in the first place. And the reason is that the aromatic ring of these molecules is well known to undergo oxidation to a quinone, and that quinone molecule is highly colored, often pink or purple. So you can see that a compound like tetrahydrocannabidiol is about a 5% loss per week at 70 degrees. And simply by taking a colorless solution of THCV that might be pure from a plant extract or a synthetic process and heating it up in the presence of oxygen, you get a direct readout on the quinone that tells you a lot about what you were starting from. And finally, we have the idea that sunlight can induce photochemical transformations. So CBC is another molecule that is very prone to a photochemical cyclization to cannabicyclol. which itself is, understudied and represents a minor cannabinoid, potentially, difficult to handle outside of the realm of a controlled laboratory setting, simply because of this variable exposure during the agricultural process. Finally, we're going to close and transition to sort of a celebration of natural products leading to life-saving medicine. And I'd like to share with you what I know about the story of Taxol. In the way of Taxol, we're going to also come up against this concept of semi-synthesis. So let me just define that we have fermentation as a process to mimic exactly how the plants make their molecules. That's where genetically modified organisms express a plant protein in water and allow it to catalyze one or more precedented steps. We've been talking a lot about organic synthesis and, again, the laboratory approach that can be controlled and safe when we think about FDA-approved medicines and going through all the rigors of the process of developing a commercial viable scale-up. And semi-synthesis, I mentioned earlier, is our ability to produce a complex target molecule by deliberately starting with a transformation on a readily abundant, naturally occurring starting material or phytochemical. And all of these definitions, more or less, have played out in the story of Taxol, because when first identified in 1967, it was from a very broad National Cancer Institute funded survey across the nation for plant and microorganism-based sources of new antibiotics and natural products. And it was found in the bark of the Pacific yew tree. I've included a map of the Pacific Northwest, so you can see that this is actually a very localized population of older, fully mature trees. In the course of about 2 decades of intensive work that was complicated because of the complexity of this natural product, preliminary clinical trials showed that it had potent cytotoxic effects and showed a lot of promise on the grounds that it was a unique mode of action, a brand new mechanism for inhibiting the growth of cancer cells. And the reason is that cancer involves cell division, and Taxol disrupts microtubules. And microtubules have to come together to form a spindle apparatus that's important for separating 2 cells during mitosis. So if you block that spindle formation, you have a very potent, tumor prevention mechanism. By 2000, it was becoming the best-selling anti-cancer chemotherapeutic agent, particularly for rare and late-stage ovarian and breast cancers. And it was at this time, unfortunately, that the supply challenge became a tremendous problem. And the reason is that paclitaxel, which is the generic form of the natural product or name, was being extracted to the point of causing a mass extinction of that Pacific yew tree. It took almost an entire yew tree and harvesting all of the bark to yield a single patient dose of Taxol. And simultaneously, there was a development in Europe. It was found through other isolation work that the Pacific yew tree, which is actually a shrub, was producing a forerunner, or a more simple abbreviated form of paclitaxel called diacetylbacsatin 3rd. And I've highlighted in blue the overlay between the small diacetylbacsatin structure and the fully elaborated form of Taxol. And one synthetic chemist working out of the University of Florida, Holden and his group, identified that semi-synthesis was potentially going to save the day and come to the rescue of the need and the demand in the clinics. And he went on to develop a 4-step transformation of this diacetylbacsatin. He patented his processes, and ultimately, with the help of Bristol Myers Squibb, which was already involved making cancer derivatives or Taxol derivatives for cancer, they co-developed this patent, and it became a commercial academic partnership that became the solution to the supply problem. So Taxol is a wonderful case where the molecule was so complex that a start to finish chemical synthesis was often never going to compete. And I just wanted to leave everyone with another semi-synthesis in the realm of the cannabis industry. And this is something that we definitely want to worry about and pay close attention to. When we isolate hemp-derived CBD, and therein lies a legality associated with farming and cultivating any hemp-derived product, there has been interest in transforming that into delta H THC, whose trace presence in cannabis itself has been known all along, but the naturally occurring amounts of delta 8 are very low, less than 1%. And it really represents an understudied molecule whose precise mode of action and its influence on one or more endogenous receptors is poorly understood. It's also something that might be metabolically very unique from delta-9 THC. Furthermore, it has been hydrogenated to HHC, or the hexahydro form, and upon acetylation, we get HHCO, which is an acetylated or esterified form of the HHC. And these molecules are now beginning to look like very potent CB1 agonists that more or less do nothing for the user beyond some type of recreational high or prolonged euphoria. And so this is where some of the idea of synthetic cannabinoids can get off track and sort of miss the mark. We do want to understand that these molecules have been known as long as the cannabis plant, but they are themselves very risky prospects, purely from the standpoint that less is known about them and more studies and clinical trials would be needed if we were interested in advancing them to the clinic as robust medicines. And I'll say more in the Q&A question. I think this is a very hot topic that's ripe for discussion, and we're going to do our best to celebrate the idea that we can derivatize what is known. We can create new compositions of matter, but it's important to realize that synthetic cannabinoids would only arrive at the level of a needy patient suffering and in need of treating a disease through a rigorous process of randomized controlled trials, FDA approvals, and doing a lot of legwork to make sure that there are no trace chemicals or residues or contaminants arriving at synthetically derived molecules. Yeah. Okay. So more time would be needed, but I just want to close with the concept of biomimetic chemistry. One of the things that we've done here at CitraChem, and we're really excited about, is falling between these two lines of biosynthesis and chemical synthesis. And we call it biomimetic in the following sense. We've actually developed or uncovered a process that begins with citral, a common monoterpene that's the essence of lemongrass, or the essential oil of something that's very safe and easy to grow at large scale. And in a single operation, we can convert that into the monoterpene you see in the middle, from which we diverge and access a number of valuable phytochemicals. Those include terpenes like terpenoline and its isomer alpha-philandrine. And we can also do the traditional terpene to polyketide merger that looks very similar to how it's done in the plant using a well-known organic reaction known as a Friedel-Crafts reaction. And in this way, we're actually building the molecules with minimal steps and maximal convergency. Convergency in the sense that we have about the same size in each portion of our building block. We stitch them together in the middle, controlling stereochemistry, and we arrive at carboxylated forms of the natural products. For example, CBD methyl ester, which is just 3 steps from this abundant terpene citral, is something that in a single step we can demethylate and decarboxylate to get CBD or THC. And we can also selectively demethylate and access in a controlled way the acid forms, which are more difficult to derive from the plant itself. So I mentioned at the start that I would make my acknowledgments as I go. Again, I want to thank the ACS, and I'm really looking forward to our post discussion Q&A. Thank you, everyone, for your time.
Thank you. That was wonderful, Dr. Kingsbury.
Last slide.
Thank you for spending your time with us today and giving us this enlightening talk. As you've alluded to, we have some questions. I'd like to take a moment to let the audience members start putting their questions into the chat so that we can get back around to them. I also want to be a little bit cognizant of our time. And for some reason, Zoom is asking me if I speak Spanish. I don't know why. Where was I? So we'll be cognizant of time, but we can go a little bit over in the service of answering some of these questions. So let us start with the following question, which is, you've talked a bit about risks associated with semisynthesis. And I'd love it if you could talk a little bit more about whether sort of these risks are really kind of inherent to the process or really due to a lack of expertise and or regulation about how these processes are done and then marketed.
Yes. So Jordan, great question. I think I'll take it in 2 stages. And I want to drive home the point that when we derive the plant phytochemicals, there are inherently downstream extensive processing and purification steps that look very similar to what we would do in the laboratory approach or the organic synthesis approach. And so the lesson is we need careful monitoring in both of those realms, in both of those activities. I think one potential advantage on the synthetic side is even if we're targeting a known phytochemical, right? We want to replicate exactly what the plant is producing. The way that we administer our reagents in a traditional set of glassware or even a stainless steel reactor setting, we know what the reagent will tend to do, if it's an oxidant, if it's an acid, if it's a base, etc. So we are already a step ahead of the game in terms of knowing what we might see as a potential byproduct of that reagent. We're typically purifying at each individual outcome. So we run a step, we purify the intermediate, make sure that its composition matches the expected outcome, and we removed any contaminants that might have been a byproduct of the reagent or a co-product of the one that we actually desired and purified out. So in a way, the logic of chemical synthesis readily translates into predictability about what your process controls will look like. And if I didn't deliver this message, it can be more complex or more, harder to control in this setting where you have all of the chemicals, including the cannabinoids, the terpenes, flavonoids, potential solvents, other contaminants derived from the soil that the plants are grown in. There can be impurities in the solvents if we're not buying very high quality or purified solvents, depending on the commercial supplier. So I definitely want to paint a picture that there's issues in both approaches, but synthesis has a way of controlling, predicting, and confirming set outcomes that we almost approach in a step-by-step manner.
That's great. That's very helpful. Sort of a related question. You discussed earlier the extraction machines and product safety. Can you talk about the complexity of the analysis of that output that you've just been referring to that's needed to ensure purity? And also sort of with regard to isomer identification, can you talk about some of the techniques that are needed to evaluate that purity that sort of go beyond standard chromatography, which is commonly used? And frankly, while you're at it, can you explain to people what chromatography is?
Sure, yeah. So chromatography is a technique, a physical phenomenon that's rooted in separating molecules based on their polarity, or how water-loving or water-fearing the molecule is. And that's a function of the presence of oxygen and nitrogen, which are more highly electronegative atoms and tend to readily lend themselves to more polarity or greater polarity. Any chromatography process is going to have a mobile phase and a stationary phase. Whether it's automated or it's human controlled, you have a media, and it's typically a solid absorbent. Think of a powder that is free flowing, and you can, for example, pour into a long cylindrical column. And then you pass your mixture through the column, and with the mobile phase appropriate for what you're trying to separate, the molecules are going to begin a drag race, if you will, to the finish line at the bottom of the column, and they're going to separate and begin partitioning apart from one another based on this physical property known as polarity. On the practical side, how do we implement chromatography into extraction processes? It is a lot harder to look at a product chromatogram when it's a full spectrum of all of the chemicals in the plant. And I think we agree on at least about 140 to 150 phytocannabinoids. There's probably well over 200, 250 compounds collectively across the three classes of terpenes, cannabinoids, and flavonoids. So the methodology and the analytical work that goes into cultivation derived extracts looks very different than single targeted outcomes in a step-by-step synthesis. And what typically needs to be done is a standardization. So if you have 300 peaks in your chromatogram, your output, and that would even be a dream scenario because that would mean that you almost separated everything. What you're going to worry about is overlap of two chemical entities that look like one, but they're right on top of one another in terms of their elution time or their finish line. And so you typically have your experimental, and then you have a standardized run where synthesis is actually interfacing this process from the get-go in its ability to deliver all of the single isomer molecules that you need in a rigorous way with surety and certainty about their composition, so that we know what all of those individual peaks are in a hemp-derived or cannabis-derived extract. So the legwork is in the level of literally looking at every single peak and confirming it with a standard. And just to do that type of throughput and processing, you either need individual phytocannabinoids already purified as single molecules from the plant, or you might have undertaken a chemical synthesis with deliberate logic and design to make the molecule in a controlled outcome in the first place. And so we have a lot of commercial suppliers supplying milligrams of these standards, and we'd like to expand the repertoire of what's known to access some of these more dangerous experimental derivatives that might be being produced out there in backyard settings.
So what you're saying essentially is, in order to purify using chromatography, you need a sample of the things you don't really know yet. Right? I mean, that is to say, you're taking this mush of cannabinoids, many of these sort of 150 to 250 individual chemicals. We don't really know what they are or how to identify them. And yet, in order to know where they're going to elute, we need a representative sample. That sort of seems like a bit of a catch-22.
Yep. We recently found a company in Italy that had spent about 3 years because they saw a little peak in the baseline of their chromatogram, and it turned out it was C4 CBD. So 1 carbon shy of the real CBD, and they didn't know what it was, and they had to develop and realize a start to finish synthesis to confirm that it was in fact C4 CBD.
Interesting. Awesome. Thank you. In an attempt to decrease dangerous products like K2 spice that you mentioned, or even potentially dangerous products like HHC and THCO acetate, lawmakers have really tried to draw a distinction between plant-derived and synthetic cannabinoids. Do you think that this is reasonable? And if not, how might they more appropriately distinguish between legitimate products and dangerous drugs?
Can I weigh in? Because I'm sitting here.
Absolutely.
You know, I think there's state by state laws that are so varied, it's very difficult to navigate. But I think what they're trying to get at is the things that we don't have experience with. Delta 8 HHC, we don't really understand what they are, how they're affecting the body. We should regulate, we should get to a point where someone's studying it and understands what that impact is. The problem that we see is the term synthetic gets thrown around too broadly. So as we've been pointing out, if you can replicate the exact molecule that the plant creates, a CBD molecule is a CBD molecule. So if it was derived in a lab, shouldn't really matter relative to the regulators. What they're really trying to get at is, what are the things we don't know enough about that we should pause and learn more about?
Absolutely, no question about it. And I think your point about their unfortunate use or misuse of the term synthetic has now kind of caught them by their own petards. Another question that's been posed is, many people believe that synthetic and isolated cannabinoids are less effective than plant extracts because extracts contain many chemicals which they feel work together, often called the entourage effect, or I prefer entourage hypothesis. What evidence is there to support this hypothesis? And does that mean that no synthetic cannabinoid medication can ever be as good as a whole plant extract?
Yeah, it's a great question. Comes up a lot. I think there's a wide array of research and outcomes that we should look at. So we shouldn't throw away whole plant extract if we can find there's benefit to a patient or any individual. But at the same time, we know there is research on specific cannabinoids, which receptors they're reacting with, and how they're reacting. And it could be that maybe we need a combination. Certainly more of a Western medicine approach to say cause and effect. We think CBG can help with inflammation. Let's test it to see if that's the active ingredient that would make a good medicine. And that's very different than a holistic approach. Understood, but there's room, I think, across the spectrum for both.
It strikes me that I don't remember where it was that Jason made this comment. I think it might have been offline, but I believe he had said that there would be value in taking these extracts and then working to purify them, but in segments, right? In aliquots, if you will, to start to see whether there are combinations that seem to interact, and so that we can. Or I think actually what Jason said, and I'm really putting words in his mouth, is that one might take a look at some of these naturally occurring extracts and then try to rebuild them with a number of synthetic pure molecules to see whether we could replicate the value of those extracts in a more rigorous and controlled fashion, which strikes me as a particularly good approach.
Yeah, and I think on top of that, if you had a whole plant extract, but you really wanted a heavy CBG because you think CBG has a particular effect, you could fortify that whole plant extract with synthetic CBG to enhance the result. That would be
Absolutely.
obviously research, but that's hypothetical.
Yeah, I mean, in some ways, it's taking this concept of semi-synthesis into sort of a semi formulation where you're using some baseline extract, but then adding additional amounts of the things that you find are particularly relevant.
Right.
So here's a question. THCV is a buzz amongst patients. What should we, I'm not sure what they mean by what should we take when using cannabinoids like THCV? Perhaps that means what do we need to know about them? How is THCV synthesized, and what potential risks or adverse effects should clinicians be aware of?
Another good question for you.
Yeah, I think I would try to answer that question by saying we know a fairly little about THCV at this point. There have been some reports that THCV might be useful as an appetite suppressant. I can say firmly that if I had a nickel for every patient who came to me and said, "Doc, give me the weed that doesn't give me the munchies," I would be very wealthy. Unfortunately, the clinical research on THCV as an appetite suppressant and as a weight reduction aid does not seem to be panning out, though I would say that data needs to be replicated considerably. But interestingly, what has been shown is decreases in cholesterol and improvement of overall lipid profile, as well as insulin resistance. And so it may turn out that while it's not as sexy as being able to be the next Wegovy, it may turn out that it is the next Lipitor, and that probably has vast health implications. But again, I think that a lot of that research is very preliminary, and we need to push for that to get done in a more robust fashion. I think this will be the last question. This is from Dr. Mark Scaldoni. And he says, non-natural isomers such as delta 8, sorry, my glasses obviously need to be reevaluated, can co-elute by HPLC in most methods, but you can use gas chromatography to get baseline resolution of the isomers formed in the acid catalyzed ring closure of CBD to make either delta 9 or delta 8 THC. I think that's not really a question, that's a statement.
Yeah, and I would certainly agree with that. When you think about some of the subtle differences in either the stereochemistry the position of the double bond. These are leading to fairly unpredictable changes in receptor affinity and, resulting bioactivity or physiological effect. And when I think about some of these techniques, they might be great on analytical scale, but for example, gas chromatography, using the volatile character of some of these compounds, it's not necessarily amenable to high throughput or scalability in the context of a new medicine. So one of the problems with the THC series in general is delta 9, the prototype or the parent itself is an oil. So when we think about how we develop our syntheses and our processes, we get really excited when we have a crystalline derivative because you can easily purify away trace contaminants in a very documentable way, in a very sort of high throughput, high yielding fashion. And so some of the opportunities that might come in a synthetic route are going to look differently in terms of your angle on your end goal relative to separating this complex mixture of full or broad spectrum extracts.
Excellent, thank you. I think you guys both touched on or brought to the fore the concept that there are probably multiple different analytical methods that are going to be necessary for us to really be able to be sure that these end products are purified.
Yeah, I think our overarching point through this is, yes, there's a spectrum of how you can receive these cannabinoids and other materials. But if we're talking about compromised patients or someone who's at health risk, on the medicine side, if you really want to create a medicine, you better know what's in that medicine is not going to harm that patient.
Oh, absolutely, absolutely. I just want to say that that Mark put into the chat box a link to a GC method, which I assume is intended for things at scale. So if anybody's interested, they should be able to copy and click that link. And with that, I'd like to thank everybody for coming today, and for Dr. Kingsbury for a wonderful talk. And with that, I think we should call it a night.
Very good.
All right, thank you all again.
Thank you.
Thanks so much, everyone, for coming. Have a great night.
Just hang on.